In the first step of DNA replication, helicase unzips and breaks the hydrogen bonds between the nitrogenous bases in the DNA molecule, thus creating an origin of replication bubble. Topoisomerase and gyrase relieve the stress off of helicase while breaking the hydrogen bonds. And the two strands of DNA are held apart using single-stranded binding proteins, as shown by dots on the outside of the strand. In the second step of DNA replication, on the leading strand, primase adds a few RNA primers to create the landing site for DNA polymerase III, because DNA polymerase III is too big to land on one or two bases on the DNA strand. DNA polymerase reads the DNA in 3 to 5', but writes a 5' to 3' strand. In the third step of DNA replication, on the lagging strand, primase adds multiple RNA primers for DNA polymerase III to continue working. But DNA polymerase III only reads in 3 to 5' and writes in 5 to 3' strand. So it goes against the overall direction of replication for each chunk, called Okazaki fragments, but generally completes the segment of DNA in the correct orientation following the arrow. In the final step of DNA replication, DNA polymerase proofreads the DNA strand and replaces the RNA primers that primase put on with DNA nucleotides. But in doing so, it rips apart the backbone that keeps the strands together. So ligase goes through and patches the backbone because they were broken, thus completing and creating two new daughter strands of DNA from one parent strand.